Hi, my name is Hannah Fischbacher, and I'm a third year sports med student at SDHS. For my sixth sports med project, I will be researching chaperone molecules and its effects on Alzheimer's disease. So, what are chaperone molecules? Chaperone molecules are special proteins in the cells that help other proteins fold correctly and function properly. They're like helpers or quality controllers for the proteins. So, preventing misfolding and fixing problems. And then also, they prevent aggregation, which basically is stopping proteins from clumping together. And then, lastly, assisting during stress, protecting proteins when the cell is stressed, such as during heat or toxin exposure. So, in order for a protein to become useful, the protein needs to be folded into a 3D shape. Protein structures are evolved to fold rapidly into their final shapes, and some proteins begin the process as the end of the protein change emerges from the ribosome, which results in a molten globule, which is the most flexible state of the protein and therefore when it is folded. However, it is important for the protein to fold properly because if it doesn't, it can cause harm to the body, which is why chaperone molecules are so important. So here is a quick video on chaperone molecules.
Misfolded or aberrant proteins are not only a waste of energy for the cell, but they are also dangerous. These aberrant proteins typically have exposed hydrophobic regions, which cause them to clump together into aggregates, which can accumulate and cause severe diseases. The cell has two lines of defense against the formation of these dangerous misfolded proteins: molecular chaperones and the proteasome. Molecular chaperones help proteins fold correctly, while the proteasome destroys misfolded proteins through proteolysis. The molecular chaperones and proteasomes compete over these proteins. The longer a protein remains misfolded, either because chaperones haven't reached it or because they haven't succeeded in fixing it, the more likely it is that a proteasome will degrade it.
So, what is Alzheimer's disease? Alzheimer's disease is a progressive brain disorder that leads to memory loss, difficulty thinking, and changes in behavior. It is associated with the buildup of abnormal proteins in the brain. Symptoms include forgetfulness, confusion, trouble with language, and personality changes. Symptoms will worsen over time, eventually affecting daily life and basic functions. So, amyloid plaques are clumps of proteins called beta amyloids, which build up between brain cells and disrupt communication between those cells. And then neurofibrillary tangles are twisted fibers of proteins called tau, which accumulate inside neurons, interfering with their functions. And then also neuron death. The buildup of plaques and tangles causes neurons to die, leading to brain shrinkage, which you can see here in the photo. A normal brain here on the left compared to an Alzheimer's brain on the right, which is significantly smaller. And then also inflammation. Immune cells in the brain become overactive, causing inflammation and further damage. And then lastly, loss of synapses, which are connections between neurons. That's what synapses is. And when that's lost, which impairs communication in the brain.
Let's discuss chaperones first. Chaperones work by binding to exposed hydrophobic patches on misfolded or incompletely folded proteins and hydrolyzing ATP. Some of the energy expended by chaperones is used to perform mechanical work, but much more is used to ensure the accuracy of protein folding. The proteins that chaperones help fold properly are called client proteins. There are several families of molecular chaperones in eukaryotes, which function in different organelles. The ones we will be discussing are HSP60 and HSP70, each of which has a set of associated proteins. HSP stands for heat shock proteins because lots of them are produced when a cell is exposed to elevated temperatures, responding to increased amounts of misfolded proteins. HSP70 acts early on in the process of protein folding, binding to polypeptide chains emerging from ribosomes where there is a chain of 7 hydrophobic amino acids. HSP70 is aided by HSP40s, and many cycles of ATP hydrolysis are generally required to fold a single polypeptide chain correctly. HSP, let's discuss.
So, what causes chaperone molecules to collapse, which was a little bit talked about in the last video? But first off, extreme temperatures. Um, and then next up, oxidative stress, and then chemical stress from toxins or pollutants. pH changes, a highly acidic or basic environment can cause chaperone molecules to collapse. And then lastly, genetic mutations. So, how can you prevent chaperone molecules from collapsing? First off, avoiding extreme temperatures. Now, this is kind of hard to avoid, but this will protecting cells from excessive heat or cold can prevent chaperone molecules from destructing. And then next, minimizing oxidative stress, maintain a healthy, rich diet in antioxidants, emphasis on vitamin C and E to reduce free radical damage. And then next, reduce chemical exposure, avoid toxins, pollutants, and heavy metals that can damage proteins. Next, maintaining a stable pH, ensure proper cellular environments by staying hydrated and maintaining balanced electrolyte levels. Support cellular health, regular exercise, and good sleep improves overall cell health and stress resistance. And then prevent genetic issues by going to genetic counseling, which may help identify hereditary risks that impact chaperone proteins. And then manage cellular stress, avoid overexertion, excessive alcohol, or smoking, which can stress cells and harm proteins. And then lastly, promote proteolysis. Um, a balanced diet and healthy lifestyle support the body's natural protein folding machinery. And that is it. Thank you.